Transcription is the process by which DNA is copied into RNA in the first step of gene expression. It begins with a bundle of factors assembling at the start of a gene, that is, a linear sequence of DNA instructions, here shown stretching away to the left. The assembled factors include an RNA polymerase, the blue molecule. Suddenly, RNA polymerase is let go, racing along the DNA to read the gene. As it unzips the double helix, it copies one of the two strands. The yellow chain snaking out of the top is the RNA, a copy of the genetic message. The nucleotide building blocks that are used to make the RNA enter through an intake hole in the polymerase. In the active site of the enzyme, they are then matched to the DNA, nucleotide by nucleotide, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, thymine is replaced with the closely related base uracil, commonly abbreviated U. You are watching this process, called transcription, in real time. Eukaryotic transcription begins when RNA polymerase II binds to the promoter region of a gene. A crucial part of this initiation process is the recognition and binding of the TATA sequence, a short stretch of DNA rich in thymine and adenine nucleotides. The subunit of RNA polymerase II that binds to the TATA sequence is called the TATA binding protein. The TATA binding protein binds to DNA using an 8-stranded beta sheet that rests atop the DNA helix like a saddle. 2 protein loops drape down the sides of the DNA like stirrups. Binding of the TATA binding protein introduces a severe kink in the DNA backbone. This kink dramatically bends the DNA helix by nearly 90 degrees and is thought to provide a signal to assemble the rest of the transcription complex at the initiation site.
Protein synthesis is a 3-step process, including initiation, elongation, and termination. In bacteria such as E. coli, initiation requires 3 small proteins called initiation factors, IF1, IF2, and IF3, as well as the first tRNA, the mRNA, and the small ribosomal subunit, also called the 30S subunit. IF3 readily binds to the small ribosomal subunit, and its presence blocks the large and small subunits from prematurely associating. IF3 facilitates the binding of the mRNA to the small subunit of the ribosome. The binding occurs just 4 to 8 bases upstream of the AUG start, where a consensus sequence called the Shine-Dalgarno sequence in the mRNA anneals near the end of the 16S ribosomal RNA in the small ribosomal subunit. IF1 binds to the small subunit at a location called the A site, where incoming tRNAs normally bind. The first tRNA, called N-formylmethionine tRNA, thus enters another site called the P site. Note that the initiator tRNA has been escorted to the P site by IF2, which is bound to GTP, a high-energy molecule similar to ATP. The anticodon of the tRNA is complementary to the AUG start codon. Once the initiator tRNA is in place, IF3 is released. With the loss of IF3, the 50S subunit can dock on the 30S subunit. The docking of the 50S subunit triggers the hydrolysis of GTP on IF2 and the subsequent release of the initiation factors. The ribosome is now ready for elongation. Elongation involves the repetition of 3 steps. First, an elongation factor called EFTU, associated with GTP, binds to free charged aminoacyl tRNAs. This complex enters the A or acceptor site. Correct selection of the tRNA complex depends mainly on codon-anticodon pairing. In this example, the anticodon CCC pairs with a GGG codon. The tRNA carries the amino acid glycine. In the second step, the ribosome's peptidyltransferase activity catalyzes the formation of a peptide bond between the new amino acid in the A site and the previous amino acid in the P site. Simultaneously, GTP is hydrolyzed and the resulting EF-TU-GDP is expelled. The third step is called translocation. An elongation factor called EFG associated with GTP binds to the ribosome. The GTP is hydrolyzed, providing the energy to ratchet the 50S and 30S subunits ahead one codon. This maneuver opens up the A site and slides the uncharged tRNA into the last site, called the E or exit site. The next amino acyl tRNA that enters the A site creates a conformational change in the ribosome that telegraphs through to the E site and ejects the uncharged tRNA. These elongation steps repeat along the mRNA. The ribosomes of E. coli can speed through these elongation steps, linking together 16 amino acids per second. Eventually, the ribosome arrives at the end of the coding region, marked by one of 3 stop codons. This stage of translation is called termination. No corresponding tRNAs exist for stop codons. Instead, a protein called a release factor, either RF1 or RF2, which has the general shape of a tRNA, mimics a tRNA and enters the A site. The release factor activates the peptidyltransferase function of the ribosome, which cuts the bond tethering the completed peptide to tRNA in the P site. Another factor called RF3 then triggers RF1 or RF2 to depart the ribosome. Finally, another factor, called ribosome recycling factor or RRF, along with EFG, binds at the A site, and the accompanying GTP hydrolysis undocks the 2 ribosomal subunits. IF3 then reassociates with the 30S subunit, preventing the 50S and 30S subunits from coming together again. The liberated ribosomal subunits are now free to diffuse through the cell, ready to bind yet another mRNA and begin the translation sequence anew.
To extend a growing polypeptide chain, the ribosome must select the correct amino acids that are specified by the messenger RNA. An amino acyl tRNA bound to elongation factor TU, EF-TU for short, enters the free A site on the ribosome. If the anticodon of the charged tRNA does not match the codon in the messenger RNA, the tRNA is rejected. The process of trial and error repeats until the correct tRNA is identified. Elongation factor TU hydrolyzes its bound GTP and dissociates. If the tRNA is correctly matched and remains bound for a long enough time, it is committed to be used in protein synthesis. The ribosome catalyzes the formation of the new peptide bond and undergoes a dramatic conformational change. Elongation factor G binds to the ribosome. Hydrolysis of GTP by elongation factor G switches the ribosome back to the state in which it can accept the next incoming tRNA.
To extend a growing polypeptide chain, the ribosome must select the correct amino acids that are specified by the messenger RNA. A tRNA amino acid complex enters the free site on the ribosome. If the anticodon of the charged tRNA does not match the codon in the messenger RNA, the tRNA is rejected. The process of trial and error repeats until the correct tRNA is identified. If the tRNA is correctly matched and remains bound for a long enough time, it is committed to be used in protein synthesis. The ribosome catalyzes the formation of the new peptide bond and undergoes a dramatic conformational change. This switches the ribosome back to the state in which it can accept the next incoming tRNA.
Like DNA, RNA strands can also pair to form a double helix. The paired nucleotide bases are packed in the middle of an RNA helix, surrounded by the backbone. The RNA helix has a different geometry than a standard DNA helix. For example, the RNA helix has a hole down the middle, and also has a significantly narrower and deeper major groove. The backbone is composed of repeating ribose sugars and phosphate groups. Unlike the 2' deoxyribose used in DNA, ribose has a hydroxyl group attached to the 2' carbon. This extra hydroxyl group influences the secondary structure and is the primary reason why an RNA helix has a different shape. Base pairing between strands is similar to that in DNA, except that adenine pairs with uracil instead of with thymine. Uracil is never used in DNA. As in DNA, a GC base pair in an RNA helix has 3 hydrogen bonds. The AU pair, like the AT pair, has 2 hydrogen bonds. RNA strands often fold into complex structures. In this stem loop structure, also called an RNA hairpin, a single-stranded RNA molecule folds back onto itself. The stem at the bottom is a classical RNA helix. Bases in the single-stranded loop, in contrast, may be unpaired or engaged in non-standard interactions. Eukaryotic genes typically contain introns, which have to be removed after transcription. Before the RNA transcript leaves the nucleus, the cell splices out the intron sequences. A few short nucleotide sequences provide the cell with cues of what to remove. The elaborate molecular machine that carries out this task is called the spliceosome. A branch point binding protein, BBP, and a helper protein, U2AF, recognize the branch point site within the intron, and an RNA and protein complex, called a SNRP, recognizes the 5' splice site by forming base pairs with it. Next, another SNRP base pairs with the branch site, displacing the bound proteins. Additional SNRPs now come into play, and several RNA rearrangements occur to break apart the U4-U6 base pairs and allow the U6 SNRP to displace U1 at the 5' splice junction. Now in position, a conserved adenine nucleotide in the intron attacks the 5' splice site, cutting the sugar phosphate backbone of the RNA. The end of the intron covalently bonds to the adenine nucleotide, forming a lariat structure. The spliceosome rearranges to bring together the exons, allowing the 3' hydroxyl group of the first exon to react with the 5' end of the other. After the 2 exons are joined into a continuous sequence, the lariat is released and degraded. All tRNAs have a characteristic L shape, with an amino acid attached to the 3' end at the tip of the shorter arm. The anticodon loop is positioned at the opposing end of the molecule and contains the anticodon base triplet. The amino acid, a phenylalanine in this case, is covalently attached to a conserved sequence, CCA, that is common to the 3' terminus of all tRNAs. The anticodon is comprised of 3 nucleotides complementary to the codon in the mRNA. The bases are exposed and are thus freely accessible for base pairing during protein synthesis. In this example, the anticodon sequence is GAA, which would match a UUC codon on a messenger RNA, specifying phenylalanine. The charging of tRNAs with the correct amino acids is carried out by aminoacyl tRNA synthetases. As revealed in the cutaway view, the complex of phenylalanine tRNA with its cognate synthetase shows an extensive contact surface that includes recognition sites for the anticodon base triplet. The tRNA's CCA end is deeply buried in the enzyme.